last one. In this last video, what we're going to do is we're going to talk about our last two modifications, post-translational modifications to protein. One of them is going to be ubiquitination, and the other is going to be oxidation reduction. All right, so when we talk about ubiquitination, what this is the addition of a ubiquitin protein moiety to different proteins. And so what this is, ubiquitin is a small protein, a very small protein within your cells, and it's a tag that can go onto the protein. And so a lot of people just think that it's a tag that tells the cell that this, whatever it's associated with, is going to be broken down because it's trash. But it can actually modify the enzyme activity as well. But really, the way that we're going to talk about it as far as exercise and as far as muscle is that when you add a bunch of ubiquitins onto a protein, that's a signal that that protein that's tagged with this long tail of ubiquitins is supposed to be degraded or broken down. All right, so the enzymes that add ubiquitin to proteins are called E3 ligases. So an E3 ligase is the enzyme 3 in the ubiquitination steps, and what it does is it ligates. So that means ligate means to glue. So it's an ACE, which means it's an enzyme, and the enzyme activity is a ligation. So a ligation is gluing, and so it's an enzyme that glues ubiquitin to proteins. You can have deubiquitinating enzymes, but these are very poorly understood. These are the enzymes that remove it, and the ubiquitin molecules are again going to be added to lysines. So one of the things that we do when we acetylate, deacetylate is we make it possible for those proteins to be ubiquitinated or not to be ubiquitinated. And also, one of the interesting things that happens is certain proteins only become phosphorylated when they're ubiquitinated. So all of these post-translationally modified proteins, all of these modifications can actually interact with each other. So the primary function that we're going to talk about, though, is targeting proteins for being broken down. And they're broken down, these proteins are targeted to be broken down in a very specific way, and that's using the proteasome, which is our big trash removal system in a lot of our cells. All right, so ones that are affected in muscle, things like the muscle ring finger protein or MRF, and this protein muscle-specific Xbox protein. These are two E3 ligases that become activated and increase when we go through periods of inactivity or periods where we have long-term protein catabolic states. And so what they're doing is they're supposed to be indicators of muscle atrophy. And I'll show you some data about that. All right, so the way that this works is very complex, but all I want you to know is that one enzyme primes the ubiquitin. It gives it off to the second enzyme E2, and the E2 is going to really hand it off mostly to an E3 protein, where it's just a channel where we take the ubiquitin and we hand it like it's a baton, and this is a relay race. And what the E3 does is it identifies the protein that's going to be degraded, and it adds it on, and you get this long ubiquitinated tail, and this polyubiquitinated protein is going to be targeted to go into this proteasome, which is this huge molecule that basically is an ATP-dependent protein. It basically is like a shredder. It's like a paper shredder. It's going to take your protein in at one side and it's going to shred it out, and it's going to give you confetti on the other side. The confetti here is amino acids so that those can be reused, but basically it's taking this protein, it's got this polyubiquitinated tail, that targets it here, you cut off the tail, the ubiquitin is recycled, and the protein is degraded. And so this was first really identified in skeletal muscle by Sue Bodine. Professor Bodine, who's now at the University of Iowa, was my colleague here at the University of California, Davis. What she showed is that when she knocked out either this protein MAFBox, the E3 ligase MAFBox, or the E3 ligase MRF, what she did is then she denervated the muscle, and what you could see is that there was a 56 or a 36% sparing of muscle mass loss if they didn't have either MRF or MAFBox. So what you can see is if you don't have MAFBox, you can't degrade the muscle, and so your muscle stays bigger for longer. Whether it's functional or not, she didn't really, the team didn't go into at that time. But it's really interesting because what it's saying is that these proteins, MERF and MAFBOX, are really important for protein degradation and really protein quality control within the muscle. So ubiquitination is really important for recycling and quality control within the muscle. All right, the last one that we're going to talk about is oxidation reduction. So again, the enzymes here are actually the oxidative metabolism. So every time you increase the metabolic state of the cell, you're going to increase the production of, and you're using oxidative energy. So you're using the mitochondria, you're going to produce more oxygen-free radicals. And those can oxidize other proteins down the line. A lot of what we're going to do to reduce proteins are other things like thioreduction, or we're going to use scavenger proteins to get rid of the or the oxide-free radicals. Okay, so but if you have an oxidized protein, you can use thioreduction to basically reduce the oxidized protein back to its reduced state. All right, in the oxidized state, cysteines within the protein will form bonds with each other, and this changes the shape and activity of the cells. So you make intramolecular or extramolecular cysteine bonds, and that can change the activity of the protein. Examples of proteins that are controlled by oxidation during exercise is the one that I can really point to is this one USF1, which is important in endurance adaptations, and we're going to talk about that a little bit. So in summary of this overall lecture on translating protein, making that protein, and then post-translationally modified, once we make a protein, its activity can be altered by phosphorylation, acetylation, ubiquitination, substrate or nucleotide binding, and oxidation. Most of these post-translational modifications, what they do is they change the shape, the location, and or the binding partners of that protein. And it also changes the activity in many cases. So it can really do four things: change the activity, shape, location, and binding partners. Kinases are the enzymes that add phosphate groups to proteins, to serines, threonines, and tyrosines, which are neutral amino acids. It makes them negative, and it causes a change in protein shape. And phosphatases are the ones that remove those. And again, all of these things can have this amplification effect, where one kinase can phosphorylate three different kinases, which can phosphorylate three, and you get signal amplification. If you, instead of phosphorylation dephosphorylation, we look at acetylation deacetylation. The enzymes that add acetyl groups are acetyl transferases that take them off our deacetylases, and by adding that neutral acetyl group to a positively charged lysine, we're going to modify the protein. You can small GTPases that bind to nucleotides like GTP or GDP. When they're GTP bound, most of them are active. When they then break the GTP into GDP, they become inactive, and it's like we talked about, it's like a timer that you can set in the cell. We're going to turn this protein on for a short period of time, but we want it to turn off. Once it's off, we need a guanine nucleotide exchange or a GEF protein to take out the GDP, and then it's going to pick up GTP from the cell. And then if we want to accelerate, we want to turn it off quicker, we can use a guanine nucleotide activating protein or a GTPase activating protein or a GAP protein. And then we talked about ubiquitination as a way to target proteins for degradation.